Hello dear friends, welcome to our next video. We are from study.official. Today's topic it's on Gibberellins, which we are continuing from the last topic. Last topics we had dealt with the auxins and all. Now we are continuing with the Gibberellins. Let's see what we shall learn today. Yes, today we shall see about the discovery, occurrence, transport and mode of action of these Gibberellins. Based on your suggestions, I have cut off this Gibberellin topic into two separate videos. In this video, let's see these topics. Let's begin. Yes, dear friends, discovery. When we come to the discovery of the Gibberellins, they were actually very well known in Asia since ancient times. Do you know, friends? In those times, especially in those Japan areas, they have found that some disease in which the rice plants were growing very tall and they were not much yielding and eliminate the seed production. This was the disease that was identified by the farmers of the Japan areas. See, rice farmers from Asia had long known of disease that makes the rice plants grow tall but eliminates the seed production. Next, in Japan, this disease was the farmers in the Japan they call them as the foolish seedling disease because the growth was there, but the seed production was not there. That's what they call this foolish seedling disease, or they name also with the Bakane disease. Also, it was also named later. There were trials to find out what is the reason why what's happening in this. Then the plant pathologists, while investigating the disease, they found that this tallness was induced by a chemical, by a fungus that had infected these tall plants. These tall plants actually had been infected by a fungus, Gibberella fujikori, that's secreting some chemical. That is the reason for this tallness, and that is the reason for the non-seed production of these seedlings. And the fungi that was the Gibberella fujikori, and the chemical they named as gibberellin that is responsible for the tallness. Later, the researchers were going on. Actually, they don't know the importance of the gibberellins at that time. They were just finding out what it is, what's the use of it. In 1930s, friends, the Japanese scientists succeeded in obtaining impure crystals of two fungal growth active compounds, but this did not reach the West until 1950. See, friends, the Japan, it was a native for the Japan. So the Japanese scientists were working on that and they obtained the impure crystals of these two fungal growth active compounds. Means the chemical that is very important for this tarnish, they tried to extract the impure crystals of that, and such research activities were done by the Japanese scientists. But friends, this was not known by the West, that is the Americans or the Europeans. They actually did not know about this research activity. That's why they did not know the importance of this. Later, what happened in the 1950? The time has passed. In the 1950, two groups were allotted: Imperial Chemical Industries at Welling in Britain and USDA, that is United States Department of America in Peoria, Illinois. They characterized this structure and named it as a gibberellic acid. They were in search of a chemical that is a plant growth hormone. These people were finding the plant growth hormone that is very much responsible for the plant. But Japanese scientists were actually extracting the crystals of a chemical that is responsible for the tallness of the rice plants. There was no much relevance, but the chemical they were working on was same. In the 1950, what these people were working was same that were worked by the Japanese scientists. They call them as gibberellins. They call them as gibberellic acid. At the same time, the scientists at Tokyo University isolated three gibberellins from original gibberellins A, that is gibberellins A1, A2, A3, The original gibberellin was A. From that, the scientist at the Tokyo University in Japan, he isolated three forms, that is gibberellin A1, A2, A3. But this GA3, gibberellin A3 and gibberellic acid were found identical. This was the point at the time when they came to know that the importance of the gibberellins, or the disease, that is Gibberella fujifera, the fungus that is causing, why the importance of this gibberellin, that is the elongation of the plant length. This all came to know from that time. And Yabuta and Sumuki, in the 1938, they named active principle as gibberellins, that is active principal compound, that is gibberellins. And as per the report of 2017, till now 136 types of gibberellins are known. Yes, now let's see mode of action. What might be the mode of action? See, you may know that the gibberellin is, as we see in the rice plant itself, the length is just increasing. And it's majorly does not increase the number of nodes, it just increase the internodal distance. And the cell elongation, that is mainly the cell elongation itself that is causing. So cell elongation takes place, and see what happens during the cell elongation. Increase in the endogenous oxygen content. Friends, look into this. Gibberellic acid does not individually responsible for the growth. Whenever a GA causing a cell enlargement, the effect is not considered to be direct. It's not directly affecting, indirectly mediated through formation of auxin, in turn responsible for the cell elongation. That is, auxin is directly performing, it is indirectly acting. The gibberellic acid is indirectly mediating through the auxins. And gibberellins has been shown to cause synthesis of amylase in barley aileron cells. In the barley aileron cells, that is the seeds of the barley, it has shown to cause the synthesis of amylase in those aileron layers. Starch are converted to reducing sugars, resulting in an increase in the osmotic pressure, causes the entry of water into cells and cell enlarges. See, starch is converted to reducing sugars. When they are converted to reducing sugar, the osmotic pressure automatically increases. As a result, the cell wall or the cell membrane becomes weakens and the water gets entry to it. That's what happens. I completed the mode of action. Now let's see occurrence. Synthesized in young leaves, shoot tip, root tip, and developing seeds. This is just a common way do these gibberellins occur. Now let's see transport. Transport of gibberellins is passive. Difference, it's not active, it is passive and non-polar. They move both in xylem and phloem, and vice versa. They move in any of them, xylem or phloem, and all. They're just active. And here, it's not specified where do they actually move. They may move, but remember, it's passive and non-polar. Thank you, friends. Thanks for watching this video. Please give your feedback in the comment section. Please do like this video, share this video, comment on this video, and subscribe to our channel agristudy.official. Thank you, friends.